So today I'm going to be talking about some of the work that we've done investigating the microbiome, which is a hot topic in both human and animal health right now, and how it impacts disease outcome in a swine model using PRRS-PCV2 co-infection. So first let's define the microbiome so everybody's on the same page. This term is used to describe basically the whole community of microorganisms that literally share our body space. So this is pathogenic microorganisms, this is symbiotic, but the more that we learn about the microbiota, the more we know that a lot of these microorganisms, the vast majority, are beneficial to us. We provide benefits to them, they provide benefits to us. This is an emerging area of study, again, in both human and animal health. We can see that the increase in publications in both humans and swine have really increased over the last decade, but primarily in the last couple of years. On the human side, this has primarily been driven by the NIH Human Microbiome Project, which allocated $150 million to understanding how the microbiome was different between healthy humans and those with certain diseases. So there's several sites on both us and our pig counterparts where the microbiota colonize. This includes the oral cavity, the nasal cavity, the urogenital tract, but the majority of the microorganisms live in our gastrointestinal tract. And in fact, these microorganisms outnumber our own cells approximately 10 to 1. These include viruses and bacteria, protozoa, fungi, archaeobacteria, and are very diverse. So there's several roles of the microbiome that they play both within the gastrointestinal tract as well as systemically. We know that they're releasing molecules that affect our blood-brain barrier. They're releasing molecules that affect our systemic inflammatory process. So that it's not just localized to the gastrointestinal tract, it's really a systemic effect. First, the microbiome plays a major role in absorption and metabolism of calories. And this is a huge role in the degradation of fiber, the metabolism of complex carbohydrates, things that we don't have the ability to metabolize on our own. They also play a role as a protective barrier, inhibiting various gastrointestinal pathogens from gaining access. We also know that this microbiome more and more plays a huge role in the development of our immune system as well as the regulation of immunity throughout our life. So for our pig counterparts here, we'll talk about the factors that impact the microbiome development, but it's very similar in humans. So the initial microbiome colonization in the gastrointestinal tract primarily has to do with sow factors. So what's the vaginal microbiome of the sow when the piglet is born? What's the diet of the sow and the subsequent microbiome of the sow? Those are the initial microorganisms that the piglet is going to ingest during delivery, and also ingest during nursing, exposure to feces, those kinds of things. We also know that the environment and the diet of the piglet play a major role in the microbiota development. The transition to solid feed is one of the biggest factors in shaping the microbiome for life of the pig. We also know that various stressful events, so processing, needle teeth clipping, tail docking, injectable antibiotics is more of an obvious impact for the microbiome, but these really impact microbiome colonization for several weeks after administration. And then inherently, pathogen exposure is particularly in the digestive tract, but really any pathogen exposure can cause a dysbiosis. And then in-feed antibiotic growth promoters will modulate the microbiota so that there's an increased ability to harvest energy. So it's important to realize all these factors that impact the microbiome, because as we realize more and more how we can use the microbiome to enhance pig performance, we know that there's several areas where we could potentially have interventions that could maximize the utility of the microbiome. So the role of these microbiome in the gastrointestinal tract in health and disease is very complex. A lot of the research has shown correlative associations, not much causation. So we have to take that into account when we look at this data, but there's been several publications that show either microbiome diversity or the actual composition, so what species are there, what phylums are there, play a role in both disease susceptibility as well as the response to infections. When we think about this in humans and see what research has been done, again, most inherently we think about how the microbiome would affect gastrointestinal diseases. So we know that Clostridium difficile has you have increased susceptibility to Clostridium difficile infection if you have lower microbiome diversity. And in fact, the leading treatment for Clostridium difficile is now transplanting the fecal material from a healthy human into a person that has Clostridium difficile infection. And that has a higher resolution of disease than any antibiotic treatment targeted towards Clostridium. We also know obesity is associated with shifts in the microbiome, and that actually obesity is transmissible by microbiome transplants. So if you take the feces from an obese person and put it into a mouse model, that mouse will also become obese. So we know it has a lot to do with the microbiota. We also know there are several diseases that are associated with the gastrointestinal microbiome that are neurologic diseases. So these include things like autism, chronic fatigue syndrome, Alzheimer's, and various other disorders. And finally, we know that the gut microbiome plays a role in several pathogenic infections of the lung, as well as some immune diseases of the lung, asthma being the most probably researched. But we know that like pneumococcal pneumonia, you have increased susceptibility if you have a reduced microbial diversity. So this is really important, thinking about the hygiene hypothesis and how we're all exposed to less pathogens, swine are exposed to less pathogens, and our microbiome diversity has gone down. And how does that affect our susceptibility to disease? So thinking about how the microbiome, we know that it affects both weight gain and immunity. We wanted to see if this translated into a disease model using PRRS and PCV2 co-infection. We know that these two viruses work together to enhance disease caused by one another. There are also two viruses that reduce weight gain in pigs. They also work together to modulate the immune response. So they both are immunosuppressive, increasing the secondary bacterial infections and also viral infections. So our first research question was really just to ask the question, are there microbiome characteristics that are associated with outcome following PRRS PCV2 co-infection? Really, are there differences on how a pig responds to these co-infection, these two viruses? We did this by using experimental design of utilizing 95 nursery-age pigs. They were all co-infected with both PRRS and PCV2 and followed for 70 days post-infection. At 70 days, 20 pigs were selected for having either the best or worst clinical outcome, and feces was collected at that time for microbial analysis. We selected these pigs by first ranking average daily gain from highest to lowest and clinical disease duration from shortest to longest. We selected 10 of the best clinical outcome pigs because they had the highest average daily gain and a complete lack of overt clinical disease, so they never showed signs of disease after co-infection. The worst clinical outcome group had the lowest average daily gain and at least 10 or more days of moderate to severe disease. We confirmed that these two groups were divergent in their weight gain throughout the 70-day trial, and also confirmed that the best performing group had a significantly higher average daily gain throughout the 70 days post-infection, making it clear that we can really divide these pigs into two populations based on their outcome after co-infection. We also wanted to confirm various other factors affecting the pathogenesis of these two viruses to assess if there were microbiome associations with both lung lesions and then overall PRRS virus replication. We first looked at lung lesions, and we've already seen a great picture, but this is normal lung, and this is a lung with severe interstitial pneumonia. So this would be a score of 0, and this would be a score of 4. And what we saw was the best clinical outcome group had significantly lower microscopic lung lesions compared to the worst clinical outcome group, confirming that the degree of interstitial pneumonia plays a major role in outcome. We also confirmed that the worst clinical outcome group had higher PRRS virus replication, shown here on day 28 post-infection, a more prolonged virus replication. We also looked at the total virus replication by calculating the area under the curve of the Viremia curve and show that the worst outcome pigs had significantly higher total virus load. When we started to do the microbiome analysis, we used the Lawrence-Livermore microbial detection array, and it has the capacity to detect all sequenced microbes, which is over, in the particular test that we used, is over 8,000 species. So the first thing we wanted to calculate when we started looking at the microbiome was diversity. And remember, we talked about there are several human diseases that are associated with lower microbiome diversity. We found a similar effect in that the best clinical outcome group had significantly higher microbiome diversity when compared to the worst clinical outcome group, indicating that microbiome diversity is beneficial for clinical outcome. We also looked at the composition of the microbiome between the two groups. Several bacteria were found at similar rates between the two groups, and that's the majority of what we found, a lot of similarities. However, E. coli, non-pathogenic E. coli, was found at a significantly higher rate in the best performing pigs. And when we took a step back and looked at overall the proteobacteria phylum, which only typically makes up about 2 to 3% of the overall bacteria in the microbiome, was actually only found in best clinical outcome pigs, indicating that the members of this phylum may be beneficial for outcome. We also found several viruses in the feces of these pigs. However, none of the viruses were detected at significantly different rates. So to summarize this first trial, we found that increased microbiome diversity and increased E. coli were associated with improved outcomes 70 days after co-infection. So this was really exciting for us because we figured that we didn't know if there would be a difference between outcome. And when we found out that there was a difference, we thought, well, is that because those changes have occurred during the co-infection period, or is it because those changes were actually prior to co-infection and could predict or predispose outcome? So that led us to the second question, to see if the microbiome characteristics present on day 0 or prior to co-infection at weaning could actually predispose or predict clinical outcome. To do this, we had 50 nursery-age pigs. They were again group housed here at Kansas State, and all pigs were challenged with PRRS PCV2. At 42 days post-infection, 20 pigs were selected for either having the high or low growth rates. However, we had collected feces from all 50 pigs because we're unable to predict outcome, and so we could go back to the fecal samples from those 20 pigs and analyze differences that may predict outcome. We did this, selected the pigs by ranking the average daily gain from highest to lowest. In this particular experiment, we eliminated the pigs that had shown any signs of clinical disease. We wanted to evaluate the subclinical effects of PRRS PCV2 co-infection, showing that they can reduce growth rate without overt clinical disease. So we selected high growth rate pigs from the highest average daily gain and the low growth rate pigs from having the lowest average daily gain. We again confirmed the divergence of the weight gain between these two groups, showing the weight gain was identical at day 0 prior to co-infection and then diverging over the 42 days post-infection. We also showed that the high growth rate pigs had a significantly higher average daily gain post-infection. We confirmed the lung lesions similarly to the first trial, that there was a trend for the high growth rate pigs to have lower lung lesions when compared to the low growth rate pigs, which was actually a little bit surprising because, again, none of these pigs showed signs of clinical disease. This is just a change in growth rate. However, you can see that the degree of interstitial pneumonia, even on a subclinical level, impacts growth. We also confirmed that the low growth rate pigs had higher PRRS viremia, shown here on day 4 and 14. We also showed that the total virus replication during the 42-day trial was significantly higher for the low growth rate pigs. Again, no clinical disease, just changes in growth rate, but they're replicating PRRS virus at a higher rate. When we looked at microbiome diversity, The first thing we wanted to do was look at the family diversity similar to the first trial. And what we found was the high growth rate pigs had significantly higher microbiome diversity on a family level. We took a step further to look at the diversity of the species detected within these two groups, and again found that species diversity was associated with improved outcome and growth post-infection, again showing that microbiome diversity is beneficial to growth and clinical outcome. We also looked at the composition between the fecal samples between these two groups and found 29 families. However, they were all detected at similar rates between the two groups. So we took it again a step further and looked at what number of species are found within these particular families, and is it different between high and low growth rate groups. And 3 families actually stood out to us as different. Methanobacteriaceae is in the Archaeobacteria, and it was actually found, the only bacteria that we found that was at a lower rate in the high growth rate pigs. So indicating that the members of this bacteria may be detrimental to growth. When we looked at Ruminococcus and Streptococcus, the opposite was true in that high growth rate pigs had these bacteria at a higher rate. So to summarize this trial, we found several characteristics of the microbiome that was beneficial towards growth rate after co-infection in subclinical pigs. Again, this was on day 0 and was predictive of outcome. So we found several characteristics of the microbiome as well as disease phenotypes in these co-infected pigs. But really, I think what stood out between the two trials was that at both day 0 and at day 70, microbiome diversity was beneficial to outcome. So the question is, how do we improve and increase microbiome diversity so that you have a more robust immune response to various pathogens in the pigs without introducing the pathogens themselves? And ultimately, how can we use the microbiome to our advantage? How can we use it as an alternative tool to both improve growth rates and improve the immune response to various pathogens? And I'd like to thank, of course, the funding agencies that help support this work, USDA, the National Bio and Agro Defense Facility Fund, as well as our colleagues at Lawrence Livermore. And we had a great group that worked together on these trials. Of course, Bob Rowland and everyone in Bob's lab, as well as our colleagues at Iowa State and Lawrence Livermore. And I'd like to just recognize Rebecca Ober. She was a great part of this, a graduate student. She's currently taking her veterinary school boards today, so we need to have good thoughts for her. But she was a big part of this project and is a great grad student. So thank you for your attention.